Okay, this is review for Chapter 18, Blood. So, a lot of the stuff we're going to cover as we progress through the chapter. One thing I want to note is what is blood? It's a connective tissue. Talked about this in the first part of the class. Remember we talked about the 4 types of tissues? Blood's a connective tissue. What's the matrix of blood? Plasma. Okay. You guys can read this. Note the bottom one. Oxygenated red is bright red. Oxygenated blood, blood that has oxygen, is bright red. Blood that has been deoxygenated is darker red. Now, blood that is deoxygenated is not, I repeat, not blue in color. I know some people think it's blue, and I can see how, because in the diagrams in the book, they always show veins being blue, at least the systematic veins. That's just to delineate it and to differentiate it. Okay, blood that is lacking oxygen is not blue. These are just some of the functions. You can read those. We talked about what the blood does, right? Passageway for antibiotics, antibodies, hormones. Okay, it has a lot of functions. You can read those on your own. Okay, components of blood. What's the take home here? Blood's about half fluid, half formed elements. Maybe a little bit more toward the fluid than the formed elements, but not that important. Just about half and half. Okay? Maybe notice a little bit more plasma. Okay, but I'm not going to say is plasma 51, 52, 53. No, just about a little over half plasma. A little bit less than half formed elements. Also know that red blood cells far outnumber white blood cells. Okay. This diagram shows what this diagram does, but just in a more precise way. Okay. This is, okay, in the top part, okay, see it has a blue panel to the left, and then pink panels on the bottom, and then tan panels above. The tan panels are the blood plasma, what's in the fluid part. The pink panels below are the formed elements. Okay, basically the same picture as before. Okay. Blood plasma, mostly fluid, but there's also some things other than fluid. There's proteins that are solubilized in that plasma. Okay, it's just not fluid. Again, plasma is not just water. There's other stuff in there as well. Okay, now we're talking about the formed elements. On that diagram below, this is what we're talking about. What we're going to talk about now is on this diagram, you see where it's that white layer and the red layer? That's what we're going to talk about in the next several slides. Those are the formed elements, or the non-fluid part of the blood. Okay, there's living cells in here, which are the red blood cells and the white blood cells, and there's also parts that are no longer living. Those are platelets. Okay, erythropoietin is a hormone that stimulates red blood cell production. This is red blood cells, not white blood cells, not platelets, but red blood cells. The hematocrit is the relative percentage of red blood cells to total volume, and we talked about when the hematocrit's high and low. Now, red blood cells are going to carry oxygen, okay? We need oxygen to make ATP in our cells, or we need it to make large amounts of ATP, I should say. So knowing that, what does that tell you about anemia? When you're anemic, you're weak, right? Well, if you're weak, you're maybe not making as much ATP as you should. If you're not making a lot of ATP, it might be because you're not getting enough oxygen to your cells. Since red blood cells are like trucks for oxygen, if you don't got a lot of those trucks for oxygen, that is part of the causative agent for anemia. Polycythemia is the other one, abnormally high hematocrit. Hematopoiesis. Hematopoiesis is the formation of blood cells And these are blood cells, white blood cells, red blood cells, and platelets, all from one magical cell that we call the pluripotent stem cell. Okay, that pluripotent stem cell will differentiate into myeloid stem cells and lymphoid stem cells, and those further differentiate. But they all come from a pluripotent stem cell. How that pluripotent stem cell differentiates into the blood type, into red blood cells, shown on the left, the white blood cells, shown on the right and center. That whole process is hemiopoiesis. Okay. Now what we're going to do is talk about red blood cells. Okay, this picture, all of these are basically the formed elements of blood. Starting from the left, we have red blood cells. Second from the left, we have platelets. And the remainder of the cells on the bottom are the white blood cells. All of these combined form the formed elements of the blood. So in the next several slides, we're going to look at the formed elements of the blood. We're going to spend a lot of slides on those red blood cells, or that erythrocyte. We're going to spend a lot of slides on the white blood cells, starting with the eosinophils, going all the way to the B lymphocytes. We've got a couple slides on platelets, but the structure of the continuing slides here is we're going to talk about all of these cells in the blood, which are the formed elements. Okay. So red blood cells. Basically, red blood cells are big old sacks of hemoglobin. Okay, that's all they are. They're just a big old sack of hemoglobin. Okay. We're always making red blood cells because they're always being worn out. We have to make them to replace them. Okay. Red blood cells are a biconcave disc. I got a picture of that coming up. This is a biconcave disc. They're not spheres. They're not perfect spheres, okay? They are disc. They're a biconcave disc. They lack an organelle, and they lack a nucleus. Okay, they are anucleated. We said this before. Red blood cells are anucleated. They don't have a nucleus. At least human red blood cells do not. This is not all red blood cells. In humans, they're anucleated. So basically, they're just a red blood cell. One of these. If you were to open this up, it's just a big sack of these things right here, which are hemoglobin. Okay? So this is a red blood cell. It has no nucleus. It has no mitochondria. And inside, there's about 250 million of these molecules right here. This is a hemoglobin molecule. If we were to parse this apart, we would see that there is iron, Fe, in the middle of this. Okay, so that is a red blood cell. Let's talk about this hemoglobin group. We're going to talk about this later on, but we need to make a note here. This is hemoglobin. On the left, hemoglobin. It has 4 peptide or 4 protein chains, and it has 4 heme groups. Okay? H-E-M-E, heme group. On the right, it's showing a large picture of that heme group. The heme group has 2 parts to it. It has an iron in the middle. See that Fe2+? That's the iron. And you see that stuff all around it? Those nitrogens and carbons and COOHs, all that stuff around it? That is what I'm going to call the non-iron heme group, or the non-iron component of the heme group. When we talk about how hemoglobin is broken down, we're going to look at 3 different things. We're going to look at proteins, which in this diagram are those blue lines. We're going to look at the iron in the heme group, which is that Fe in the middle. And then we're going to look at the non-iron part of the heme group. That's that yellowish part on the right that has all the nitrogens and carbons and the ring-like structures. Okay? So what do we know about these red blood cells? The what? Red blood cells, these are for transportation of oxygen. Okay? They're going to do a lot of other things, but for the most part, red blood cells are going to transport oxygen. They make ATP, but they do it anaerobically. Okay, they don't have any mitochondria to do aerobic respiration. This is not to say they can't make ATP. They can, but they do it through anaerobic pathways that do not need a mitochondria. What I was talking about before, hemoglobin has two parts: a globin, which is the protein, and the heme group. Now that heme group has an iron part and the non-iron part. Okay, we talked about that before. Now I talked about how red blood cells are formed, right? How they're formed and then broken down. So what's some take-home points here? Well, first of all, this shows how 3 parts of the blood are dealt with. Okay. Let's look at the number 1. We see red blood cells, right? This is, we're looking at number 1, this large pink blob. This is a spleen, a liver, someplace where the red blood cells have gone to die. And we said that what? Red blood cells are made up of what? Red blood cells are made up of the globin, which is that blue part made of protein, and the heme group in the center. And that heme group has an iron part and a non-iron part. So we're concerned about 3 sections here: the protein, the iron in the heme group, and the non-iron in the heme group. And this diagram shows the fate of those 3 components. Okay? So let's start at number 1. A red blood cell comes in. It's worn out. It's been about 4 months circulating. It's worn out. Its day is done. It has 3 components. It has the globin, which is the protein. It has the iron part of the heme group, and has the non-iron part of the heme group. Okay? All 3 of those are broken down in separate pathways. Now we're going to look at that globin first, that protein, those blue lines on that previous diagram. Well, that protein is made of what? Amino acids, right? We're going to take those amino acids and break them up. We're going to take that protein and break them up into the amino acids, and those amino acids are then recycled. Okay, they'll do something else in the body. They're like blocks, right? They're building blocks. Okay, so we've taken care of one thing, the protein of that globin protein. Now that leaves us with the heme group, which has what? An iron part and a non-iron part. Okay. Steps 4, 5, 6, 7 talk about what happens to the iron part of the heme group. Steps 9, 10, 11, 12, and 13 talk about the non-iron part of the heme group. Okay, and that diagram from before, remember the iron was that orange dot in the center, and the non-iron part was all those cyclic carbons and nitrogens surrounding it. Okay? What do you need to know about what happens to iron? Well, first of all, iron is going to combine with transferrin, basically like a truck or a carrier, and it takes it to the liver. The liver is like a storage house for iron. From the liver, it's going to be stored until it's needed, right? And then it's going to, when you need something, you warehouse it, right? You collect it and you store it in a place until you need it. The liver is like a warehouse for the iron. At some point, you're going to need to make red blood cells, right? And you're going to need the iron, which is an important component in this. So when there's a need for iron, you know, in the red bone marrow, iron goes from the liver, shown in step 7, to what? The bone marrow, where the red blood cells are made, shown in step 8. From the bone marrow, what happens? Red blood cells are made. It combines with different components, and that iron is just one part of the larger piece of the puzzle. and eventually it makes red blood cells from new hemoglobin, and that new hemoglobin circulates throughout the body for about 4 months. Okay. But what's the take home here? Starting from the left, we break a red blood cell down. We eventually scavenge, get one of those irons from the hemoglobin in the red blood cell. We take that iron and store it in the liver. We then, when needed, take it from the liver to the red bone marrow. We then make a new hemoglobin from the iron from before. That new hemoglobin is one among hundreds of millions in a red blood cell. A red blood cell is formed and then released and circulates for about 4 months, and then that red blood cell is worn out and goes back to the spleen, for example, and that is broken down once more. Circle of life, guys. Okay. The other part here, we've talked about what? The protein, the iron part of the heme group. That leaves us with the non-iron part of the heme group. Just know that it also goes to the liver, to the small intestine, large intestine, where bacteria breaks it down and it's released as feces. Okay, but here's all the steps to that. Okay, and you know this gives feces its color. All right, let's move on. Erythropoiesis is the formation of red blood cells. Hematopoiesis is the formation of blood cells. Erythropoiesis is the formation of red blood cells. Difference there. There is a difference. Okay, so we're done talking about red blood cells structurally. Now we're going to talk about the blood types of red blood cells. Okay. What is on the surface of a red blood cell? What makes up blood type? Well, the first thing is the ABO blood type system. Now, this is one system among many systems out there. Okay, most well known, but this is not the only blood type system out there. Okay, so what do we know? Let's go to this picture right here. If you have type A blood, your red blood cell makes an A antigen. It has a tag. Think of an A tag on it. B-type blood has a B tag. AB blood has both an A tag and a B tag, and type O has neither A tag or B tag. You also have antibodies that will target those tags. Okay, so if you have type A blood, you have an anti-B antibody. So if a B antibody or a B-type blood came into your system, your anti-B antibodies would attack it. Okay. If you're type B blood, you have anti-A blood antibodies. That dark pinwheel, that's a IgM antibody, by the way. That dark pink, purple pinwheel thing, right? Those are antibodies. Let's just say you're type B blood. Your blood has B antigens or B tags on them. But that antibody will find and target type A tags. And when it finds them, it's going to target the cell with them, attack it, and set up an immune response to destroy that cell. Okay. If you're type AB, you have both of those tags and you don't have any of the antibodies. And if you're type O, you have neither of those tags, but you have antibodies against them. Okay. This kind of says the same thing. You should know who the universal donor is, who the universal acceptor is, and what type of blood someone with type A and type B can take. If you're type A, you can take type A blood and type O blood. If you're type B, you can take type B blood and type O blood. If you're type AB blood, you can take type A, type B, and type AB, and type O. If you're type O blood, you can only take type O blood. You should know the blood donation rules. Okay, RH, people have are you RH positive, RH negative? This is a primary medical concern when a female is RH negative and she becomes pregnant and the father is RH positive and the fetus is RH positive. The fetus will make blood with this RH factor. The female will then make antibodies against the fetal blood. Not that big of a concern, during the first pregnancy, but it becomes a major problem during the second pregnancy because the female has built up an immune response to the RH of the blood. When that baby makes its blood, which is RH positive, the female's immune system will attack the child because it is making what it perceives as an antigen or a foreign body. Transfusions, we talked about those. The third part's important. If you get incompatible blood, what happens is hemolysis. Your blood splits open. It rips it open. This happens to the new blood that comes in that's not compatible, and it can happen to existing blood. Not as much, but it will happen. So for example, let's say you're type A blood, and you're given type B blood. Well, all that type B blood is targeted, right? And it will lyse, it will break apart, your body will break apart the B type blood. And in that immune response, it can actually target some of your type A blood as well and cause hemolysis there as well. Not to the same extent as B, but it'll still happen. So know what happens if you give incompatible blood. Hemolysis. The blood is targeted by the antibodies and it's ripped apart. Now what we're going to do is talk about white blood cells. Okay, we've talked about red blood cells, and now we're talking about white blood cells. Another part of what? The formed elements of the blood. So what do we know? White blood cells loosely are the leukocytes. Okay. A couple things, they're nucleated, they have a nucleus. They're classified as being grainy or granular or agrandular. This appears with staining. Okay, this granularity appears with staining, and it's often things like vesicles of lysozyme, things like this, that cause that graininess. Again, that graininess is from the stains. Now, all of these white blood cells have specialized functions. Okay, but they don't function in a vacuum. They don't function as separate entities. Their functions are interlocking, which is to say the function of one leads to the function of another one. Okay, it's like a well-oiled machine with cogs and gears, right? When one moves, the other one moves. That kind of idea. You do not need to know these examples for the test. I just put them here to show that what? Each of these white blood cells, when one functions, it starts the other one functioning. Okay. White blood cells, very few in the blood. Not to say there's very few white blood cells, it's just they're mostly in the lymphatic system. Okay? White blood cells are mostly in the lymphatic system. There's not that many in the blood. When there is elevated amounts of them, it's a problem, actually. There's probably some type of infection. Okay. And then that last point, we'll talk about that later on. So now we're going to talk about specific white blood cells. Okay, going back to this picture right here, you see how we have the eosinophils and basophils, all these. We're going to talk about each one of these separately. Neutrophils makes up most of the white blood cells. First to arrive at the site of injury. Okay, it digests bacteria. We're talking about that in the next slide. And then, when you have an infection, you get pus. What happens is these neutrophils come in, do their thing, they wear out and die at that site of injury, and they build up. And a lot of that pus that you have is actually dead neutrophils that have, died. done their duty. Okay, so how do neutrophils work? Neutrophils work by phagocytosis. They're going to be like vacuums for bacteria, in a sense. They're going to suck those bacteria in and other pathogens and chop them up and destroy them. Lifetime is about 10 to 20 of these phagocytotic events. It can suck in about 10 or 20 bacteria. After it's sucked in 10 or 20 bacteria, it's done. Okay? The machine has worn out. The components inside have worn out, and that neutrophil dies. Basophils, another type of white blood cell. How are these going to do? They're going to go to the site of injury in the capillary and cross over. This process is called diapeptesis. Okay. At the site of injury, they release histamine, and when they release histamine, it dilates the blood vessel. So it makes the blood vessel wider. That blood vessel is made up of cells. So as it makes that blood vessel wider, it's going to cause very small little holes to form in that blood vessel and make it, in a sense, leaky. Okay. This causes, when in a localized area, an immune response, an inflammatory immune response. Now, when you have a hypersensitivity to something, when you have a massive dump of histamine, what happens? Well, when you have a dump of histamine, all the histamine is released at once. Or not all of it, but a large amount of histamine is released at once. A lot of your blood vessels become leaky, and clinically what happens is your blood pressure crashes. It goes down tremendously. Because what? All the histamine has released, or a lot of histamine has released, and when a lot of histamine has released, a lot of blood vessels have become dilated and leaky, and just like water pressure in a pipe, when there's holes in a pipe, water pressure goes down. When there's a lot of holes formed in your blood vessels, guess what? Blood pressure goes down. Same idea. Your blood pressure crashes. That's what happens when you have a large release of histamine due to some type of hypersensitivity or anaphylactic type shock. Okay. But key pointer, basophils release histamine. Basophils release histamine. Eosinophils. Key points here. Eosinophils, they're going to phagocytize their target, okay? But the important one with eosinophils is the second one. They are going to spew their digestive enzymes around their target. Okay? Why is that important? Well, it's important because, you know, we're talking about bacteria and viruses, things like that. That's all good and well because these cells are larger than those. But what happens when your target is much bigger, right? Like an intestinal worm. Well, eosinophils are good for that because they're essentially surrounding these worms and spitting on them with their digestive enzymes. Okay? So think of these little guys surrounding this big, like, worm from Dune. I don't know if you guys saw the movie Dune, and these big old land worms moving around. Well, these little eosinophils are like these little people, like, you know, throwing rocks at him and attacking him. One or two eosinophils can't really hurt them, but if you got millions of eosinophils surrounding one of these worms, that mass action of releasing these digestive enzymes around these intestinal parasites, that will help get rid of them. Okay? An important point here, when you see an elevated eosinophil level and a differential white blood cell count, it implies that you have got a parasitic infection. Okay? Okay. Monocytes, another white blood cell. Okay. These are large, and they do phagocytosis. Okay, and they're like the cleanup crew, essentially. Think of monocytes as the cleanup crew after the event has been finished. Okay, lymphocytes. Lymphocytes, this is an umbrella term in a sense, and it's going to cover B cells, T cells, and natural killer cells. Now, we can't differentiate these based on staining, which is why we call them lymphocytes, because staining will identify them to the lymphocyte level, but more testing is needed to determine if they're T cells, B cells, or NK cells. So in the next few slides, we're going to talk about B cells, T cells, and then NK cells. Now, I can't give a lot of information here because next semester in A&P, you talk about the immune system, and you'll spend half a class, if not a whole class, talking about B cells. And the same thing with T cells. And I'm just introducing the cells here. You'll get a lot more on these cells down the road. Okay? This is just a minimum amount of information I have to put out about them. You'll get a lot more on these later on. B cells, what's the key point here? These are going to release antibodies. B cells release antibodies. That antibody immunity is called humoral immunity. Okay? But these cells release antibodies, and those antibodies travel in the blood. T cells. T cells have to go and actually get close to their targets. Okay, they can't just make antibodies. T cells do not make antibodies. T cells attack infected cells. Okay? How are they infected? Sometimes there's a virus inside the cell, all right? Another thing is transplant cells. We have MHC markers on our cells that identify our cells as self. If you get an organ transplant, you have to take immunosuppressive drugs, because if you don't, your T cells will see that your new organ has different self markers on it, and it will attack that cell. Okay. An autoimmune disease is when these cells, they attack self cells, and they do not detect them as being self cells. Okay. Natural killer cells, similar in like T cells, but they use a touch kill mechanism, but they are another type of lymphocytes. White blood cells, lifespan varies. Plasma cells, they're not going to live that long. Memory cells, they're going to live for years. Now, what memory cells and plasma cells are, discussion saved for BY264. Just know their lifespan varies based on what they're doing. Okay, we talked about how white blood cells move. They're going to move through diapeptisis. When they cross over the blood vessel, that's pretty much it. But just know what diapeptisis or immigration is. I always called it diapeptisis. Okay. White blood cell counts. You should know one. Okay, let me start by saying this. You might have heard on television shows where they say run a CBC, perhaps. A CBC is a complete blood count. Now, a CBC with differentiation or a differential CBC, these look at your blood and then they identify the proportions of the different white blood cells we covered. You know, what percent are neutrophils, what percent are basophils, what percent are monocytes. And these numbers are informational because an elevated level of one or a low level of another one is indicative of a particular disease. There's a lot that the book mentions here. For example, if you have a lot of neutrophils, it might mean a burn or bacterial infection, right? What you're responsible for is this. When you have a parasitic infection, you likely have high eosinophil levels. Leukemia is when your white blood cell count is low. That's the only ones that I think you should know. Okay? Finally, platelets. These come from a megakaryoblast. And the platelets are going to be used to stop bleeding. Hemostasis is the formation of a blood clot or of a block that will stop bleeding. Don't worry about knowing all the 3 steps. Just know that hemostasis is when bleeding stops by the formation of a plug inside. We talked about the picture here, but just know what's happening is you're making a plug in a sense, and that causes the stopping of hemorrhaging and bleeding. If it was not for this, you would hemorrhage out. People with hemophilia don't have this ability, and they continuously bleed. And that wraps us up for the test on this chapter.